I'm Professor Johnson, and in this video we're going to be looking at some concepts in genetics that go beyond just the basic Mendelian. We're really going to start by expanding on some of the basics of Mendel, looking at pedigrees and test crosses. So these might even fall into what Mendel was actually working on, and then actually expand beyond that and look at some of the things that he really never grasped or even really thought of with his experiments. So pedigrees, you may know, are sort of like genealogy charts, where you're looking at a trait and seeing how it's being expressed in multiple generations. Pedigrees are especially common when you look at genetics in humans. So it's not really ethical to take two humans and say, I want you to breed, and then we're going to go ahead and count your offspring, and then we'll have your offspring breed, count their children, and determine the type of inheritance. That's not ethical. You just can't do that. So it's easier to take a pedigree, look at the history of a trait in a family, see how it's been inherited, and determine a good guess for how the inheritance of that trait actually happens. So this is a pedigree over here where the squares count as or represent males and the circles represent females, and the shaded individuals are the ones that actually express the trait. So you can see here the original dad actually had this trait. He passes it on to one of his sons, but of his three other children, none of them get it, although one of his daughters does end up having a son of her own, so one of his grandchildren actually express this trait. And so this trait's actually probably a recessive trait because it skips this generation, or this daughter here, so you look at it in this generation and it skips it. So that means that this dad probably passed on the trait to this daughter. This individual was probably heterozygous for this as well, and so when these two had children, this person got two of the recessive alleles and expressed it. Now the mom here probably is recessive or heterozygous for this as well, because she would have had to donate an allele in order for this individual to express it. So we don't know about this son and this daughter, whether they're well, they would have to be heterozygous for this because they would have gotten the trait from their dad and not from their mom. So in this case, it's probably a heterozygous one because it skips a generation. If it's a dominant trait, you typically see it in every generation, and if someone's expressing it, it's very likely that one of their children would be expressing it as well. The second thing we're going to be looking at is a test cross. So a test cross, let's say that you have an individual that shows a dominant phenotype and you want to know whether their genotype is homozygous or heterozygous. So you can really discover this by using a test cross. You're going to take this individual where you don't know the phenotype and you're going to cross it with an individual that is homozygous recessive for this. And you're going to end up with one of two different possibilities. If your individual is homozygous and has both two alleles for the dominant trait, in the subsequent generation that's been crossed with this recessive allele, all of their children are going to show the dominant phenotype because they're going to be heterozygous. However, if the individual originally was heterozygous, when you crossed it with someone that is recessive for the trait, you're going to end up with individuals that are showing, or you're going to end up with two possibilities. You're going to end up with either the dominant phenotype or the recessive phenotype, because you're going to have individuals that are either heterozygous for this because they're going to inherit the dominant allele from this individual, or they could inherit the recessive allele and then be the recessive individual because they're going to inherit the recessive trait from the other parent that was recessive. So in this case, you're going to have the 50-50 ratio or 1-to-1 ratio of dominant to recessive alleles. All right, looking at a few concepts beyond Mendel, we're going to start with polygenic inheritance, where one gene or multiple genes can affect one trait. Then we're going to look at pleiotropy, where one trait can affect multiple, or one gene can affect multiple traits. we're going to look at the case where you have more alleles than you actually have, possible alleles than you actually have room on your chromosomes for. So there's more alleles than you can actually inherit. We're also going to look at the cases of codominance and incomplete dominance, two very similar ones, and hopefully I'll explain the difference to you. And then we're going to end with looking at epistasis, how one gene can affect the expression of another gene. In the next video, we'll look at linked genes and look at a specific case of sex-linked genes as well. So let's get started with polygenic inheritance. So polygenic inheritance is when you have multiple genes. So you have the A gene here, the B gene, and the C gene. But they're all affecting the same trait. So common traits that show polygenic inheritance are height and weight, for example. So you can inherit a variety of different genes here that affect your height. So you can see two individuals that are of average height have these two different alleles for gene height. When you cross, there's going to be 64 different possibilities of combinations for this height gene, and 1/64 of them are going to end up being very tall because they get all of the alleles that bump up their height, but 1/64 are going to be very short. And then you'll end up with more of these intermediate ones, especially a lot of the people in the middle that were about the height that you were. So with this, you usually end up with somewhat of a bell-shaped curve, and that's what you see when you look at height in a population. There's a lot more people that are of medium height than are especially tall or especially short, and a lot more people that are of moderate weight than are especially heavy or especially light. All right, so the other case where you have one gene affecting multiple traits, so think of a gene that could affect your height, your weight, and maybe your bone structure or something. So these are multiple traits that are all being affected by one gene. So this gene has multiple effects throughout the body that leads to these different effects, and that's what pleiotropy is. Now, multiple alleles. You can only inherit two alleles for any one gene because you're only going to have two chromosomes, and that gene's only going to be, and you can only get one copy on one of those chromosomes and another copy on another chromosome. But there's always the possibility that another allele could be out there that you just couldn't inherit. So this is what multiple alleles are. The common example would be the ABO blood type. So you can have the A allele, the B allele, or the O allele. And so that's how we end up with the A, B, and O blood types. Blood types are also a good example of codominance. So codominance occurs when you have both traits being expressed in an individual. So the A and B blood types are codominant to each other and are dominant over the O blood type. So what the ABO blood type is coding for are these cell surface markers on the surface of cells. So if you have these markers, the cell recognizes that as, hey, yeah, you are me. But if you are of a different blood type that doesn't have one of these markers typically on there, and then one of the cell with these markers comes in, your cell is going to recognize this as foreign and will start attacking it. So individuals that are AB will have both the A marker, that's what's shown here in the blue, and the B marker normally on them. So if someone with an A marker or a B marker or both the AB markers donated blood to these individuals, these blood cells would look identical to the AB blood type, and so these people could accept them. People with the A blood type, so getting two of the A alleles is the same as getting an A allele and an O allele, because the O allele is just the failure to express one of these. These people can get blood types from anybody with the A type because it will recognize the blue marker as normal, but it couldn't get it from AB because these green markers would be viewed as foreign to these individuals, and they couldn't get it from B. They could get it from O because O doesn't have any markers on there, so there's nothing to identify it as foreign. B is going to be similar to A, except that you're looking at the B marker instead of the A marker. And then O blood types, these are the universal donors because they have nothing to mark them as unique on them, so they can donate it to any of these blood types, but they can only accept blood type from other O individuals. Incomplete dominance is very similar to complete dominance, except that instead of a phenotype where you're expressing both of them, so with codominance AB, you are both A and B. You're not intermediate between A and B. It's a absolute thing. You are A and B blood type. You can get the A blood type, you can get the B blood type, so this isn't really an intermediate one, it is a dominant one where you're showing both of them. Incomplete dominance, you're somewhere in between. So you could really not call either of these dominant to each other, although commonly the red one, because it's more red than white, the pink flowers that you end up with, can be viewed as that. It's not completely dominant. You end up with an intermediate phenotype. So this is pink instead of being completely red. So if you were to cross the pink flowers, then you'd end up with a 1:2 to 1 ratio, both phenotypically and genotypically, because you'd end up with 1 red flower, 2 pink flowers, and 1 white flower. Finally, this brings us to epistasis. So epistasis is where you have one gene that can affect the expression of another gene. So we'll give an example where you have different varieties of Z maize, corn. And these varieties have different colors of kernel, either white or purple. So you have one variety of white corn and you cross it with another variety of white corn and you end up with a purple corn. And then you cross the two purple corns and you end up with this weird ratio, 9:7. So what's going on here? Well, if we look at this, we're looking at two genes. So this is a dihybrid cross, and you're looking at the A gene and the B gene. And this person is homozygous recessive for the B, so that makes them white. This is homozygous recessive for the A, and that also makes them white. But the purple color is actually the dominant color here, even though it had two recessive parents. So if you're heterozygous for both of these, you can express purple. So these are two heterozygous individuals. You can see anytime these individuals have both at least one dominant A gene allele for the A gene and one dominant allele for the B gene, they end up being purple. So all of these have at least one, sometimes two, of the dominant alleles for both of the genes. However, anytime you have a recessive individual in either or both of the genes, you end up with the white. So this individual is recessive in the B, this one's recessive in the B, this one's recessive in the B as well. These three are all recessive in the A, and this one's recessive in both of them. So how this is working, let's say this is a biochemical pathway, and you have X being expressed. X can be converted to Y, and Y can be converted to Z. Now A and Y are both white products. Z is a purple product that gives you its purple color. If you have the dominant alleles, these are expressing enzymes. So remember, big concept here is the idea that genes are coding for proteins. Some proteins are enzymes. In this case, these are enzymes that are converting these different products into subsequent products. And the end product here is Z. So if you have an active A, either one or two of them, you can make Y. And if you have an active B, either one or two of them, you can make Z, but only if you can get Y produced first. However, if you have the recessive allele, that means that this A gene is not active. And then if you have two of those, so two A's, little a's here, you couldn't take X and convert it into Y because these are recessive genes. It's not functioning. B is working the same way. So if you have two recessive of these, that means you inherited an enzyme that's not functioning, and you can't take Y and convert it to Z. So any individual here that has the dominant A, but two recessive Bs can make Y, but that product's still white. It can't make the colored product of z. And if you have an individual that's recessive in both the a's, it can't convert x to y. And if you can't convert x to y, you never have y to make z, and so these individuals being 1 as well. All right, so I hope that was useful for you, and I will talk or see you next time.